There is an entire civilization operating beneath your garden beds right now, and if you are not working with it intentionally, you are leaving enormous amounts of plant health and yield on the table. Mycorrhizal fungi are among the most ancient and powerful allies a gardener can cultivate. These organisms have been forming partnerships with plant roots for an estimated 450 million years, long before flowering plants or humans ever existed. Research published in the journal New Phytologist confirms that over 80% of all land plant species form these fungal partnerships, making mycorrhizal relationships the rule of plant life on Earth, rather than the exception. Understanding how this relationship works is one of the most transformative things you can do for your living soil garden. The word mycorrhiza comes from the Greek words for fungus and root, and that etymology tells the whole story. These are fungi that live in direct physical union with plant root systems. The two most relevant types for home gardeners are arbuscular mycorrhizal fungi, or AMF, and ectomycorrhizal fungi. Arbuscular mycorrhizal fungi penetrate directly into root cells and form branched structures called arbuscules, which serve as the primary exchange sites between the fungus and the plant. These are the fungi most vegetables, herbs, flowers, and many fruit trees associate with. Ectomycorrhizal fungi, by contrast, form a dense sheath around root tips and are the primary partners for most conifers and many hardwood trees like oaks, beeches, and pines. Knowing which type your plants need helps you choose the right inoculant when starting seeds or transplanting. The most immediate and measurable benefit mycorrhizal fungi provide is a dramatic expansion of the effective root surface area. Fungal hyphae, the fine thread-like filaments the fungi extend into the surrounding soil, are between 2 and 5 micrometers in diameter. For comparison, most plant root hairs are between 10 and 20 micrometers. This means fungal hyphae can penetrate soil pores, aggregate spaces, and microhabitats that plant roots cannot physically access. A landmark study, published in Plant and Soil, demonstrated that mycorrhizal associations can increase the nutrient and water-absorbing surface area of a root system by anywhere from 100 to 1,000 times. That is not a rounding error. It fundamentally changes what is available to your plants. These hyphae can extend several centimeters to over a meter beyond the root zone, essentially acting as a long-distance supply chain. In a single teaspoon of healthy living soil, you can find hundreds of meters of fungal hyphae. When you hold a handful of rich garden soil and notice those fine white threads running through it, you are holding part of this network in your hands. Phosphorus is the nutrient where mycorrhizal fungi most visibly change the game. Unlike nitrogen, which moves through soil water with some mobility, phosphorus binds tightly to soil particles and essentially stays where it lands. Plant roots rapidly deplete the phosphorus immediately surrounding them, creating a depletion zone that the root cannot move past. Fungal hyphae explore far beyond this depletion zone and access phosphorus deposits that would otherwise be completely unavailable. Research from the University of Western Australia showed that mycorrhizal plants can take up between 4 and 10 times more phosphorus than non-mycorrhizal plants grown in identical soil conditions. This is not a small improvement, it is a categorical shift in what the plant can access. Beyond phosphorus, mycorrhizal fungi also facilitate the uptake of nitrogen, potassium, calcium, magnesium, zinc, copper, and iron. Zinc and iron are particularly important trace elements that are often limiting in high pH soils. The fungi produce enzymes that convert organic nitrogen and phosphorus forms into plant-available mineral forms, essentially acting as a biological nutrient processor running constantly in the root zone. This is why living soil systems with active fungal networks often support robust plant growth with far less added fertility than conventionally managed gardens. One of the most practical benefits of mycorrhizal associations for the home gardener is what happens during dry spells. Because fungal hyphae reach deep into soil microstructure and access water held in spaces root hairs cannot enter, mycorrhizal plants maintain turgor and metabolic function at soil water levels that would cause measurable wilt stress in non-colonized plants. A meta-analysis published in Mycorrhizae, reviewing over 60 independent studies, confirmed that mycorrhizal colonization consistently improves plant water status during drought. The mechanism involves both the physical access to deeper water reserves and the regulation of plant water transport proteins called aquaporins, which the fungi influence through chemical signaling. This same resilience extends to salt stress. Gardens in areas with high mineral content, groundwater or soils, or those where de-icing salts have accumulated, often show sodium toxicity symptoms. Research published in the journal Environmental and Experimental Botany found that mycorrhizal colonization reduced salt-induced oxidative stress in plants by increasing antioxidant enzyme activity and reducing sodium accumulation in shoot tissue. This is particularly relevant for gardeners working in coastal regions or reclaiming land that has been under conventional agricultural management. The physical presence of fungal tissue around and within root cells provides a mechanical barrier against soil-borne pathogens. Organisms like Fusarium, Pythium, and Phytophthora, which are responsible for damping off root rot and wilt diseases, find it significantly harder to colonize roots that are already occupied by mycorrhizal fungi. The fungi also produce antimicrobial compounds and trigger the plant's own systemic resistance pathways, effectively priming the plant's immune system. Research from Wageningen University demonstrated that tomato plants colonized by Glomus mossiei showed significantly reduced severity of Fusarium crown and root rot compared to uninoculated controls, even under conditions designed to promote disease pressure. This disease suppressive function is one reason that diverse biologically active living soil systems tend to experience fewer catastrophic disease episodes than sterile or conventionally managed soils. When you build a living soil ecosystem with genuine fungal diversity and activity, you are building a layered immune system for your entire garden. One of the most remarkable and honestly underappreciated contributions mycorrhizal fungi make is the production of glomalin, a sticky glycoprotein that, believe it or not, was only identified and characterized by USDA soil scientist Sarah Wright back in 1996. Glomalin is secreted by arbuscular mycorrhizal hyphae as they grow through the soil, and it coats soil particles, binding them together into stable aggregates. These aggregates create that crumbly, open structure that gardeners often describe as great tilth. Soil aggregate stability determines pore space, which in turn governs water infiltration, air movement, and root penetration. Research published by the USDA Agricultural Research Service found that glomalin can account for between 27 and 37% of the carbon stored in stable soil aggregates. It's also remarkably persistent, with a half-life estimated at 7 to 42 years, depending on soil conditions. This means every season of active mycorrhizal growth is contributing to soil structural capital that pays dividends for years to come. When you add compost, reduce tillage, and inoculate transplants, you're investing in glomalin accumulation. That physical improvement in soil structure is something you can actually see and feel in the texture of your beds over successive growing seasons. This relationship is not one-sided. The plant doesn't receive all these benefits for free. In exchange for the expanded nutrient and water access the fungi provide, the plant transfers a significant portion of its photosynthetically fixed carbon to the fungal partner. Depending on the study and crop species, plants channel between 10 and 20% of their total photosynthate to mycorrhizal fungi. This is exactly why the relationship is called mutualistic rather than parasitic. Both organisms benefit in measurable ways that increase their survival and reproductive success. From a gardening perspective, this means your plants are already operating as partners with the fungal network, and all you really need to do is create conditions that support and maintain that partnership rather than disrupting it. High phosphorus synthetic fertilizers are among the most damaging inputs to mycorrhizal networks. When soil phosphorus is artificially abundant, the plant has no incentive to maintain the carbon cost of feeding a fungal partner, and colonization rates drop sharply. Research from the International Rice Research Institute found that high phosphorus fertilization reduced arbuscular mycorrhizal colonization in rice roots by over 70% compared to unfertilized controls. Tillage physically severs hyphal networks and disrupts the spatial architecture that took the fungi considerable energy to construct. Fungicide application, even broad-spectrum biofungicides, can reduce or eliminate fungal populations entirely, and bare soil with no plant cover leaves the fungi without a host and without carbon flow, causing them to decline between seasons. Protecting your mycorrhizal networks means reducing tillage to the minimum necessary, avoiding high-phosphorus synthetic fertilizers, keeping living roots in the soil as much as possible through cover crops or perennial plantings, and being selective about any fungicide applications. When you do start new beds, or transplant into areas with disturbed soil, using a quality mycorrhizal inoculant applied directly to the root zone at planting gives the relationship the best possible start. Look for products containing species relevant to your plants, like Glomus intra-radices, and Rhizophagus irregularis for vegetables and annual crops, with viable spore counts confirmed right there on the label. If this guide changed the way you think about what's happening beneath your garden, there's an entire community of growers doing the same work at Living Soil Garden. Subscribe to the channel for evidence-based, deeply practical content about building genuine soil life, growing nutrient-dense food, and working with nature instead of against it. Share this guide with a fellow gardener who's ready to stop treating soil as just a growing medium and start treating it as a living system. The underground network is waiting.